to drug device combination products. We're fortunate to have some notable speakers. We're going to lead off with Tausif Ahmed, who's the Senior VP and Head of Clinical and Biopharmaceutics in Mankind Pharmaceuticals. And he's going to join us and talk about challenges with drug device combination products. DDCP, by the way, is the abbreviation for drug device combination products. And he'll talk about clinical conduct and quality from the industry perspective. And then we'll have Dr. Shinae Kim, who's a biomedical engineer from my division. She'll be talking about ophthalmic drug device combination products comparative analyses, followed up by Commander Andrew Fine, who is a senior advisor in the Division of Clinical Review in OSCE OGD. And he's going to talk about drug device combination product and its submissions and some lessons learned. And he'll also talk about some of the basic premises behind the device constituents for the ophthalmic drug products. And then the last speaker will be Ajay Khopade, who's the Vice President of Sun Pharma. And he's going to talk about challenges in the development of complex generic ophthalmic products overall. And that'll be followed by a panel, an expert panel. I'm covering because Nathan Reed is unable to make it here from St. Louis, but I understand he will be joining us online, and he's been dialing in to listen to us converse here in this workshop. So we'll look forward to conversations that we have at the workshop as well. So we can start off with Tausif is here. Hello, welcome. Up in steps. Yes. Big one.
Hi, good afternoon. Welcome back after the lunch. I think the advantage towards the end is that most of the things, the context is set. Some of the slides may be redundant, so I'll try to skip those slides in the interest of time. At the outset, I would like to thank CRCG for giving me this opportunity. I'm fortunate this is my kind of third time I'm coming to CRCG for different presentations, so fortunate and always privileged to be here and learn new things. So today I'm going to touch base on challenges with the ophthalmic drug device combinations, focusing primarily on the clinical aspects. I think during the last day and today we have talked about the options of waiving of the studies, but looking at the challenges, how we can have a mix of preclinical and clinical complemented with these approaches is what I'm going to touch base with. As a typical disclaimer, these views are solely mine and don't represent my organization, which is Mankind or Lifestar Pharma. So during the next 20 minutes, I'm going to touch base on some of the introduction, some of the challenges in terms of the ocular dosing capacity and why precision is very important, especially from a clinical perspective, and in terms of the ophthalmic drug design, then some of the emerging solutions which are coming in and focusing on the clinical conduct of these ophthalmic studies, some of the case studies which I've taken and how we could take an approach and looking at maybe an alternate approach which could help get the generics early in the market. I think one of the critical aspects in terms of ophthalmic drug delivery systems is that the volume of the accurate dosing is something which will drive the safety and efficacy. And typically we see that eye capacity is somewhere around 10 to 15 or maximum 20 microliters. But when we look into the conventional dosage forms, we'll see they will dispense somewhere between 25 to 70 microliters. And there is always some wastage, and larger drops, they will not add to the efficacy. And the key design factors which will have an impact on the clinical outcome are nozzle diameter, squeeze pressure, the droplet size, and surface tension and viscosity. And as we see that the consequences of the oversized drops could be that if we have a variable dose volume, drop volume, it could lead to inconsistent dosing. It could lead to overdosing, leading to some of the systemic side effects, and underdosing could lead to reduction in the efficacy. And all this will also have some economic factors that we will, for the long-term treatment, it will have an impact on the actual usage and the cost of the medication. So the solution to all this is precision delivery for accurate and reproducible dosing. I think these are the general aspects when we kind of look into the challenges of the ophthalmic product designs. We have to look into the physicochemical factors such as the surface tension, which impacts the droplet formation, the corneal spreading, and the viscosity, which affects the ocular retention and drug absorption. In terms of the clinical impact, the dose consistency has an impact because it affects the comfort, the bioavailability, and if the droplet size or the medication is such that it has a higher residence time, it may lead to better absorption, but if it is too high viscous, it will also lead to discomfort, blurred vision, and if it is too low viscous or the viscosity is too low, it will have a reverse impact, which is on the rapid drainage and poor absorption. And all these are the challenges which we encounter when we're doing the clinical studies. So what are the benefits of an optimal design of a drug-device combination? So we have to, in order to reduce the dosing frequency, we have come up with solutions of developing gels or sustained release dosage forms. And for better tolerability, there has to be a balance between surface tension, tearing, stinging, and enhanced precision is the key to all these problems. As we said, I think when we are designing these dosage forms, we have to consider the patient. We have to always keep patient in mind, and patient can be both geriatrics, pediatric, and patients which have different ways of handling the medications. So squeeze force and dexterity is one of the important aspects which impacts the clinical outcome. The drop dispensed depends on the bottle rigidity. See, if the patient is elderly or of younger age, the dexterity will also have an impact, and they may struggle with consistent pressure. And studies have shown that there could be 7x to 4x variability in the bottle designs, which ultimately will have impact on the clinical outcome, and this is because of the inconsistent dosing of the dosage forms. I think when we talk about these clinical studies, they are long-term, either 3 months, 12 weeks, or 6 months, and during this duration, they're always the self-administration of these dosage forms have. So, what are the impact of the patient self-administration on the clinical trial outcome? The most common factors which lead to dosing errors include the incorrect head tilt or the droplet angle or blinking during instillation. These are all practical challenges. The bottle tip contact, which will lead to contamination. And there has been reports that up to 80% of the patients use improper techniques, which ultimately have an impact on the clinical trials and the efficacy and assessment of the outcome of these trials. And these will lead to the skewness of the data, the reproducibility of the data, and the variability in terms of getting an inconsistent response. Now moving on, some of the quality and manufacturing challenges. I think we have heard a lot of this, but focusing more on the clinical aspects which we would cover. The maintenance of sterility is one of the critical aspects in terms of development of drug-device combinations. The material compatibility in terms of leaching, the adsorption, the degradation. So the selection of the right material is another critical aspect which needs to be considered during the development of these products. And we have to meet, without saying, the quality aspects for both the drug as well as the device. So both go hand in hand. And one of the major challenges which we face during industrial development of these products is the the manufacturing consistency. See, many times we heard about the polymer change. Sometimes the vendors, they change the device, the material gets changed. And when those things get changed, they have an impact on the product performance in terms of the sterility, in terms of the droplet size. So these are some of the aspects which need to be kept in mind when we are developing these products and maintaining consistency in the product life cycle. I think one of the other aspect, I think we also have this topic covered in the subsequent section, is the human factor studies. This is another challenge which industry faces in terms of conduct of the human factor studies, where I think there is no very clear-cut guidance. There is a guidance available, but then the study design, the sample size, the factors to be assessed, all these are the gray area. And we need a lot of interaction with the agency, and we would seek further guidance and further clarity to be provided in this area, where the human factor studies, there are a lot of questions which come from the regulatory agencies when these studies are being submitted. So this is an area which requires certain additional maybe clarity or more detailed discussions to happen. In terms of the bridging studies, I think this point I'll cover in the subsequent slide as well. And the other aspect is in terms of the global harmonization. See, when we are developing a product globally, the requirements for different regulatory agencies vary, and as a product development organization, it becomes a challenge to meet all the requirements. So I think a lot of efforts have been made towards this direction, but global harmonization is another area which requires further attention. I think how we work, or what are the best practices in industry where we are trying to mitigate these risks. One is obviously having an integrated teams. We involve the engineering teams, obviously the formulators, the procurement teams, and the regulatory teams right upfront in the drug development when we start. And we ensure or try to mitigate all the points which we had mentioned, and still we see those gaps, but those are part and parcel of development. I think what we have seen, and it's very heartening to see that agency is very open for early regulatory engagement. I think this is an area, especially for the clinical development, for the implants. I think some of the products guidance have come recently, and I have those slides in the subsequent slides, which give a lot of clarity, and now the focus is on the IVRT. But achieving an IVRT, having a discriminating ability, that itself is a big challenge. So the model which we would like to propose is maybe an alternate approach, where we would do the IVRT as per the PSG. If we find differences, and those are bound to be there because of the complexities we discussed in the morning, I think we want to propose a hybrid approach where we would do preclinical studies and also do a small bridging human study. I think that is something which we want to propose, and we will come to the agency with our proposal, that we will have in vitro IVRT data, Q1, Q2 data. If we are seeing differences, to justify those differences, I think we heard about if those differences are within the design space, still the gray area is there. But to kind of bridge that gap, we would like to propose doing preclinical PK studies and also human bridging studies, which may not be fully powered, but kind of a pilot studies to give kind of a totality of evidence approach. I think another area is the precision dosing, which is a very critical factor in the development of these drug device combinations, and we have faced challenges in the clinical studies which we have done, where we see a lot of variability, and those can be avoided by using some of these already approved technologies like OptiJet, which kind of delivers exactly 8 microliter, which is in line with the vitreous humor, the capacity, and there is no wastage or there is no subtherapeutic impact. It is there. So these are some of the areas which are upcoming. And there is also, in terms of the digital and patient support, when we are talking of AI and everything, the cloud-based monitoring of daily adherence and compliance of the self-administration by the patients is another area which is regularly being used in the clinical studies. I think we have talked about this, and I'll talk in detail of those two, the ones in the next slide. So I'll maybe. So I think in terms of improving the patient adherence, what we'd like to focus is to have sustained release implants, which we are talking today in this session. Those regimens have to be simplified so that it is beneficial even for the visually impaired patients. And enhanced delivery is another area which could improve the patient compliance and ultimately can lead to the benefit to the patients. And there have been a survey where the non-compliance rate in glaucoma has been reported to be as high as 59%. So when we develop these implants, these issues of clinical non-compliance can be avoided, and these are especially useful for long-term outcomes and cost-efficient for chronic therapies like glaucoma. I think I briefly touched base on these considerations in the trials which we do, and you would be wondering that why I'm focusing on the trials. See, the reason I'm trying to focus on these trials is that there is definitely a possibility that if we are not able to meet the Q1, Q2 or the IVRT requirements, we will come up with alternate approaches to the agency. And when we come up with alternate approaches, which is a mix of both preclinical as well as clinical studies, these would be challenges which we would be facing. And we would like to understand how we could mitigate this, such as the trial design complexity, integration of the dual endpoints, the training of the patient, which is extremely important for the consistent and reliable dosing, the blinding challenges which are there because of the subtle device differences, and the device management in terms of non-compliance or malfunctioning of the device during the trials. Yeah, I think I'll move to the case studies, and just to give you a perspective that by doing so, we talked about this product, and Durista is one product which is Metoprost sustained release. It's been approved by agency, but what were the challenges which were faced? Again, the challenges faced during the clinical conduct of this were the sterility validation, which requires rigorous aseptic processing. Packaging validation was another challenge due to intracameral administration. The patient usability, it required a special clinical setting and requiring ophthalmologist training and patient education. And then the regulatory classification, and the outcome was that it definitely, the product was approved, but then the path which the organization or the sponsor passed through in terms of the conduct of the clinical trials is a big learning for the future development. I think another example which I would take is Travoprost. I think for this also very recently the PSG has come, and this requires, I've taken an example where a clinical study was done for ocular implant with two different types of elution, fast eluting and slow eluting. And just to give you the context of the clinical study, I mean, it was like almost done in 600 patients. It was a non-inferiority trial where they had used a fast eluting, slow eluting, and timolol as a standard of care. And the primary outcome was a change in baseline in the intraocular pressure at day 10, week 12. Finally, the primary endpoint was week 12, as well as the safety outcomes. And this data says that the mean reduction for the fast eluting was 6.6 to 8.4 mmHg. For the fast eluting, it was 6 to 8.5, and it was pretty much in line with the timolol group, and it was found to be non-inferior. Even the adverse event profile was more or less similar. I think the overall thing which I wanted to show or kind of tell from this is that now I think even for this product, the product specific guidance has come, and it kind of recommends doing an IVRT. But what I want to trigger the thought here is a hybrid approach. So we may not do this extensive PD study or clinical endpoint study, but are there thoughts, or can we look into an approach which we call as a alternate approach, where we do Q1, Q2 sameness, IVRT, we do a PK in preclinical species, and a PD, which is a kind of a bridging study, or we call it as a pilot study. It may not be powered with 600 patients, but maybe a study in a small number. I mean, that number could be 40 patients or whatsoever, and then use that approach to kind of validate our models, as well as to kind of justify some of the differences which are within the design space. I think that's the thought process, or that's the thought which we have, and we are working on it. And I think I got very good insights in our yesterday focus discussions, that there are different platforms through a CC, or we could approach as a pre-ANDA meeting. And I think we will come back on this approach. I think the last case study is, we have talked enough on this product, but again, the concept is the same. Here, the endpoint is in terms of efficacy. This is for diabetic macular edema. And again, this study was a phase 3 study where it was done against placebo. It was for a 3-year duration with a follow-up, and this is for 6 months treatment. And the primary endpoint was a greater than 15-letter gain in a best corrected visual acuity. So this was, I mean, for a different indication, and the endpoint was different. And the outcome was that we could see a 22% with this group as against 12% with sham, which was statistically significant, and there were rare or manageable AEs. I think I don't know, these slides somehow, I don't know, somehow they missed, but no, I think the key point which I wanted to mention here, I had sent the updated slides. Somehow some of those slides look like missing. But anyways, I'll kind of cover. The key message which I want to give, even in this case, is that, is there a possibility where we could work on a model where we do maybe a preclinical rabbit PK? I think we heard a lot, but rabbit PK alone may not be sufficient. And then we talked about, can we substantiate it with in silico modeling? But the challenge we face is we don't have data to validate. And when we submit a data, then the obvious questions from the agency, which is but obvious, that how do you validate this model, or how do you do the translation? So is the next wave, or is the option of working on a hybrid approach where we do the in vitro sameness, preclinical species, and a small bridging, a PD study in a small number of patients, supplemented with a modeling? Can that be an approach be used as a totality of evidence and an alternate approach to the existing approach? And looking at the challenges which we discussed, can this be looked into? I think this is my last slide. And I think what I have just discussed in the last few slides are that precision delivery is the key area in terms of getting the enhanced clinical outcome, and to have better patient adherence. And we have to combine precision, convenience, and improved outcomes. I think some of the challenges which the industry faces is in lack of regulatory pathway in some of the areas, especially when there is subtle differences, and when we are not exactly matching and not exactly Q1, Q2, Q3, what is the way forward? And that's where we are stuck up. How do we manage the device differences between the generics and the RLD? I think the human factor studies also have its own limitations and challenges, which we have seen for a couple of products. And I think the way forward, or some of the success factors, could be obviously approach agency or early engagement with the agency, so that we can get a clear way forward, and integration of the drug device in terms of safety, efficacy, and patient compliance, and balance the clinical benefits with the usability, and also to address the supply chain management issues, devise the mitigation strategies right early in the development. I think with this, I would end my presentation, and thank you, and look forward to further discussions during the panel part. Thank you very much.
Okay, good morning, good afternoon, good evening, all the attendees virtually and in person. My name is Shinae Kim. I'm the pharmacokinetist from the Office of Research Standard within the Office of Generic Drug at the FDA. So today I'm gonna introduce the comparative analysis framework for the ophthalmic drug device combination product development. So I'm gonna start with a disclaimer. This presentation view is reflect my own, and any references, including the marketed product in this presentation, is for the illustrative purpose only. So this is a brief outline of my presentation. So by end of my presentation, I hope you can understand the comparative analysis framework for specifically for the ophthalmic drug device combination product. And you can apply best practices for evaluating user interface differences based on the current FDA guidance. And you can also learn how categorize user interface differences and how utilize the pre-ANDA consultation opportunities to enhance your ANDA submission. Sorry about it. So I'm gonna start with several key definitions, which is frequently speaking in my presentation. So drug device combination product, which called the DDCP, is a therapeutic and diagnostic product, which include both drug conjugate part and the device conjugate part, as defined in 21 CFR 3.2e. And the user interface is all component of the product which a user interacts with. It is moving. I don't know why it is automatically moving. But anyway, it includes a device control part, obviously, and also any associated controls, displays, product labeling, and packaging. And critical task is a user task that if it is not performed at all or performed incorrectly could harm to the patient or end users. And external critical design attribute is a design feature that directly affect how users perform a critical task that is necessary in the drug delivery. Okay. So the ophthalmic drug device combination product has several different presentation. However, the most common presentation is a dropper or dispenser, including single dose vial with a dropper tip or multi-dose bottle with a dropper tip as shown in the figure. So if you have a product which have this type of container closure system, that it will fall into the type 2 drug device combination product, as it functions as a pre-filled drug delivery system. And of course, we can see the insert and implant in the ophthalmic drug device combination product. However, its classification is really case by case. Applicator co-packaged with the product or preloaded system is obviously served as a device control part. However, for the inserter and the implant itself can be classified differently based on the product. So if you are developing the complex insert or implant product, you can check the PSG first, whether it is classified as a drug device combination product first. However, if you cannot find that information in the PSG, you can submit the control correspondent to the FDA. Here is another example what is not considered a drug device combination product in the ophthalmic product. So this is an example of the Tobradex ointment, which is supplied in a tube with a polyethylene tip and cap. And as you can, based on this labeling, the small amount, about the ½ inch of the drug, should be applied in the V pocket between the eyeball and the lower lid. However, as you can imagine, the tip can deliver the ointment to the V pocket. However, it does not control the specific dose. The delivered dose will be determined by the users. So the tip And the tube in here is considered as a container closure system, but not the device control part. However, this example does not mean all ophthalmic ointments are not considered as a drug device control part. So if there is any uncertainty or you need a clarification of your product, please submit the controlled correspondence. The base of the regulatory fundamentation of the generic product is a therapeutic equivalence framework. So same expectation is applied for the generic drug device combination product as well. So your generic product should be deliver the same clinical effect and the safety profile compared to the RAD. Especially for the user interface perspective, we expect the generic combination product have to be used without the intervention of the healthcare professional or without the additional training when it is substituted for the RAD product. However, it does not mean that the generic product and the RAD should be identical in terms of the user interface. So as long as the design difference does not preclude the approval for the ANDA submission, then some level of the design difference can be accepted. So this slide brings us the core message in my presentation about the comparative analysis framework. So if your product is classified as a drug device combination product, then you have to submit the comparative analysis as a part of the ANDA submission. Comparative analysis have to include 3 components: physical comparison between the RAD and the generic device consumption part, comparative task analysis, and labeling comparison. I'm gonna introduce more detail in the following slide, but before we move on, I would like to highlight here that your comparative analysis must be performed between your generic product and the RAD, not the reference standard or other approved generic product. Physical comparison includes the visual, auditory, tactile examination of the physical features of the proposed generic product compared to the RAD. It means you have to include the comparison in size, shape, color, transparency, any feedback, texture, sound, thickness, font characteristics also could be included. And you can also include all components necessary to the device drug delivery, including the packaging and the connector as well. Also, in the physical comparison, you have to include the side-by-side clear color photographs for both RAD and the proposed product. And once you completed the comparison, you have to provide a clear design differences with a clear justification for any design differences that you noted. Second component is a comparative task analysis. This analysis is about the comparison step-by-step user process. So from the opening the packaging to the disposing the product, you have to include all steps comparison between the RAD and your proposed generic product. And you need to highlight the design differences in task, especially for affecting to the critical task. If any design difference impact the existing critical task, or if it created a new critical task, you have to highlight the design differences in the comparative task analysis. As with the physical comparison, you also need to provide a clear identification, clarification, and of any design differences, and we also need to provide the justification. And I also like to highlight that this comparative task analysis should be focused on the task rather than the labeling comparison. So some of the forms sometimes submitted the IFU comparison under this session. However, it is not expected from the FDA. The last component is the labeling comparison. In the labeling comparison, you have to use the current version of the RAD labeling. So for the pre-ANDA evaluation, we normally focus on the IFU comparison, the instruction for use only. However, in the ANDA submission, you have to include the side-by-side, figure-by-figure comparison of all labeling components. So generic product labeling should be the same as that of the RAD, except for the permissible differences described at the 21 CFR 314.94884. However, in general, permissible design differences in the user interface falls within the scope of permissible labeling differences. So once you complete the three components of the comparative analysis, your outcome should fall into one of the three classification. The first one is a no design difference. It does not need an explanation. The second is a minor design difference. If you find any design difference between the RAD and your proposed device, and it does not affect any external critical design attribute, then you can classify the minor design difference. However, if there is the design difference, it may impact the external critical design attribute, then you can classify its other design difference. And when you make an outcome of the comparative analysis, please consider any identified design difference in the context of overall risk profile. So when you find the other design difference in your comparative analysis and the justification alone is not enough to support your design difference, FDA normally recommended two options. The first one is you can consider the modifying the device design to minimize the design difference compared to the RAD. The another option is you can provide additional data or information such as comparative user human factor study, in vitro study, published literature, or alternative approach. However, the type of the information and data should be, I'm sorry, it is automatically keep moving. Should be depend on the design differences and its potential risk. So information you submitted with the other design difference have to support the design that other design difference will not change the overall risk profile when it is when your product is a substitute for the RAD product. So in this case, we are highly encouraged to recommend. Highly encouraged to submit the control correspondent or pre-ANDA meeting request to discuss your approach is appropriate for the ANDA submission. And when you find the other design difference, we are highly recommend to consider the context of use and the patient population. In the product, for the RAD product, there is a different use. So whether it is emergency use drug or the routine use drug, how often that product could be used, who is the end users, and what is a patient age group, and what is the use environment. That factors will impact the decision of overall risk profile. And you also need to consider the patient population as well. So dexterity, age-related limitation, cognitive availability, disease-specific impairment also influence on your decision. So please consider these factors when you're assessing the other design difference. So this is the best practice for your comparative analysis. So when you perform the comparative analysis, please identify all user interface differences first and classify any differences based on the definition in the guidance. It means you have to classify no, minor, or other design differences. And also, if you find other design difference, please provide a clear justification. And you also need to focus on the potential differences of the critical task. So please remember that not every task is a critical task. And you also need to consider the overall risk profile based on the context of use. So even though there is a same design difference, it can be classified differently based on the RAD. So you need to tell, your comparative analysis need to be tailored individually based on the RAD product. And FDA product-specific guidance is an essential resource. You can find the current agency thinking and what is the expectation for the generic product development. So as you can see on the right example, under the additional information section, there is two device and user interface related section is included. So inside the device section, you can find what is a RAD device characteristics and which part is a device construct part of the RAD. And we also highlight that the specific device features you need to examine when you develop the generic version. And in the user interface assessment session, you can find the comparative analysis and FDA reference, FDA guidance. You need to find it to reference. And before I wrap up, I would like to highlight the pre-ENDA program again. This program is to clarify the regulatory expectation early in the product development, so it can support the applicant in the developing more complete ENDA submission. So eventually, it will promote a more efficient and effective ENDA submission, and it can reduce the number of assessment cycle for your approval. And if you developing the complex ophthalmic drug device combination product, please, you can submit the controlled correspondence or pre-ENDA meeting request for the comparative analysis evaluation with a specific question. So I think this is my last slide. This is a key takeaway and the strategy for your success in the user interface assessment. Please start user interface assessment using the comparative analysis early in development and minimize the design differences when it is possible. And if you find any other design difference, then document it thoroughly with clear justification or scientific evidence. And you can engage FDA early through the pre-ENDA program. Before I wrap up, I would like to highlight again, the generic drug device combination product are not required to be identical in the user interface compared to the RAD. FDA has accepted some level of the design differences if there is no change in the clinical effect and the safety profile. I hope this presentation helped to clarify of your comparative analysis development, and I'm gonna thank you so much for your attention.
Good morning, good afternoon. I guess we have people joining from all over the world. That's great to see. All right. So my name is Andrew Fine. I'm the Senior Advisor in OGD's Division of Clinical Review within the Office of Safety and Clinical Evaluation. Our group's primary responsibility is to evaluate the comparative analyses that are submitted in ANDAs, and that of course includes ophthalmic drug device combination products. So I'll walk through our experience with these products and share some lessons learned in these ANDAs. Here's my disclaimer for any marketed products or examples that are shared, these are for illustrative purposes only. All right, for the next 15, 20 minutes or so, in 15 minutes, everyone should be able to accomplish these objectives, which is to review the user interface considerations for ophthalmic drug products, discuss examples of the user interface assessment in ophthalmic products and ANDAs, and discuss lessons learned for ANDAs of ophthalmic drug device combination. So Dr. Kim introduced a lot of the framework and principles for comparative analyses. I'll just do a quick review of some of these and apply them to the ANDAs that we've seen in our division. So all ANDAs require apply to the framework of therapeutic equivalence, which is that an ANDA can be expected to have the same clinical effect and safety profile when administered under the conditions specified in labeling. These same expectations apply to drug device combination products, and we expand this to include that a generic combination product, we consider whether the end user can use the generic combination product when it is substituted for the RLD without the intervention of a healthcare provider or additional training. These products do not have to be identical to the RLD, but as long as those differences don't preclude approval in an ANDA. While the comparative analysis is the framework, the regulatory context is important for each of these ANDAs. The comparison is always going to be to the individual RLD. And all relevant determinations of sameness under a 505J are made respect to that RLD. And when RLD information is unavailable, performing the comparison to the RLD may be challenging, but it's still required. So what is the landscape of ANDAs and comparative analyses and user interface assessments that we've seen in our division? Since 2018, our division has seen over 1,300 ANDAs, and this pie chart breaks down the types of products we've seen according to route of administration. From this pie chart, in routes of administration for injectable products, so IV, IM, sub-Q, for example, and orally administered products make up over half the ANDAs for user interface comparisons. However, after that, a large chunk, 18%, are ophthalmic products. And I'll walk through a lot of these examples today. Dr. Kim did a nice job introducing the comparative analysis guidance. I'm not going to go through it in detail, but this is the 2017 guidance that outlines the framework for comparative analysis assessment under ANDAs for drug device combination products. There are three main comparisons that are done in an ANDA: physical comparison, the comparative task analysis, and the labeling comparison. And Dr. Kim did a great job breaking down each of these comparisons and what the expectations are. But just to highlight the types of differences you can see when comparing an ANDA to its RLD, there can be no difference, or if there are differences present, they can be classified as either minor differences or other design differences. The key distinction between these two different classifications would be whether that difference affects an external critical design attribute of that proposed product, and really using those definitions of what a critical task implies or what a critical task defined as Dr. Kim shared a few moments earlier. Just a few reminders about when we assess these differences. All difference should be assessed, adequate justification for that design difference classification, and really the focus should be on those potential differences in the critical task between the RLD and the drug device company. And of course, consider context of use. Just as a reminder, I saw a few photos of the context of use slide from Dr. Kim, and here's just a reminder. Well, when we talk about context of use, which is the key component of assessing those differences, we're referring to the urgency of use, whether it's emergency or non-emergency, the frequency of use, it's a single-use product or a repeated chronic use product. Who is that end user? Are they a patient or caregiver, or are they a healthcare professional? What is the environment of use? Is it a clinical setting or an at-home setting or a school setting? And lastly, there are patient population characteristics that factor into context of use. Some examples here are if the patient population or end user population have dexterity issues or maybe incapacitated. So now I'll walk through how we approach user interfaces for ophthalmic drug device combination. The key examples that we've seen in ANDAs are for those dropper dispensers. 800 regs define that eyedroppers are a medical device, and if they're co-packaged with a drug, those can be drug device combination products. We primarily see the multi-dose vials and bottles. We see single-dose preservative-free vials depicted here on the bottom right. Again, when these are packaged with an ophthalmic drug product, these are considered combination products. In general, when we talk about the ophthalmic user interface features, relaying back to some of the general definitions and framework we use when we talk about comparative analyses, usually ophthalmic products do not include instructions for use. They're usually a patient or caregiver administered, but there are exceptions. And the context of use is often chronic or acute use. Rarely would these be emergency. Ophthalmic user tasks, specifically for these commonly seen multi-dose dropper bottles, are to remove the product from the carton, remove or break the tamper-evident safety feature, remove the cap, squeeze the bottle to deliver the prescribed number of doses or drops to the affected eye or eyes, and lastly, replace the cap. I'm sure we've all used a multi-dose bottle. But as you see from these case studies, it's not as simple as just opening a bottle and administering the medication. So this first example, when we see differences in the tamper-evident safety feature. Tamper-evident abbreviated here is TE. Here we see an RLD product on the left in each panel, which uses a shrink seal, plastic seal as a tamper-evident feature. And on the right, an ANDA with a retained plastic ring on the bottleneck, which would be broken from the cap after first use, and that plastic ring is retained on the bottle when you administer the medication and as well as when you with repeated use. Sorry, I just want to focus on the bottom right there with that beige tamper-evident ring that's retained in the bottleneck. And this type of difference is oftentimes acceptable, and I'll walk through how this assessment is made. Here's an example of another tamper-evident ring for bermudinine tartrate. The drug product review in this example determined that the proposed bottle cap design was not acceptable because that tamper-evident ring was loose. The difference in the tamper-evident feature may impact the safety profile of the proposed product because the quality and functionality of the TE ring cannot be confirmed, and this type of difference would be not acceptable. The focus on the tamper-evident plastic ring and the user interface assessment, as well as the quality assessment, dates back to 2016 when there was a drug safety communication where FDA warned consumers about potential risks of using eye drops packaged in these bottles with a loose safety seal. There were several reports of ocular injury when inverting these bottles, the plastic ring actually fell off and caused injury. As a result, the safety communication educated the public that the plastic safety seal or tamper-evident ring should stay connected to the bottleneck, and that FDA strongly recommends when using these rings that the bottle cap design includes a positive retention mechanism. So how do we apply this to our user interface assessment and take into account those two examples I shared just a few slides earlier? The tamper-evident ring security is a quality issue and primarily assessed by the quality and the drug product team in our Office of Pharmaceutical Quality. Applicants need to certify in writing that the ring is secure and will not pose a safety concern to patients. From a user interface assessment, we need the confirmation as well in order to make our conclusion. If there are quality concerns identified, then the user interface is impacted and the comparative analysis outcome is inadequate. Just want to make note that there is a lot of overlap and collaboration with quality teams. Other studies like drop size comparison, dose accuracy, those are not part of the comparative analysis or user interface assessment. Those are primarily assessed by the quality. So based on all this information, how do we how do we break down our user interface assessment for tamper-evident ring differences? One, as most of these products do not contain instructions for use. The tasks are self-evident, and the cognitive steps in performing the tasks are similar. The end user does not need additional training if there's differences in the tamper-evident ring, and this does not impact a critical task. Next, I'll move on to examples of cap color that do impact the user interface. Here is an example of prednisolone ophthalmic suspension. As you can see in the panel on the left, the RLD labeling describes the prednisolone product as having a pink polypropylene cap. This is consistent with the American Academy of Ophthalmology's AAO's recommendations that anti-inflammatory steroid products include a pink cap, just like the RLD has. Sorry, the timing. In this situation, we have an ANDA on the right with the before and after. This ANDA initially proposed a white cap. After an information request and request to change it to the pink and align with the AAO as well as the RLD, the applicant modified their cap to match the pink, and this is an acceptable difference. So these color codes for topical ocular medications are not a new aspect of ophthalmic ANDAs as well as NDA products. So the American Academy of Ophthalmology endorses the uniform use of a color coding system for the caps and labels. With input from industry and FDA, the AAO developed a uniform color coding system. This helps patients and healthcare providers distinguish among various medications and minimize the risk of errors. There's been exceptional voluntary cooperation and compliance across industry and the agency. So I gave the example before of the pink that's associated with anti-inflammatory steroid products. There's many different colors that are listed based on the class of products. For example, tan, which we're seeing in the first example, is the color associated with anti-infectives. And yellow is associated with beta blockers. All of these are available on the link from the AAO website. My last example includes a healthcare administered product and actually is with a sterile dropper bottle. For this product, with the RLD on the left and an ANDA on a hypothetical ANDA on the right, the RLD has a sterile dropper bottle. RLD labeling states that you open the package using an aseptic technique, and to do so, users must tear between the blister packs, as you can see with a tear on the RLD picture. After opening, the bottle is allowed to fall upon the sterile surface and can be used. In this situation, there are differences between our ANDA and the RLD. The first difference includes the perforation in packaging. The RLD blister packs are perforated while the ANDA is not. The consequences of this could be increased risk of contamination and wasting of the drug product. If using the ANDA as substituted for an RLD, without the perforations, the user may tear and inadvertently open the neighboring packaging, which that adjacent product would have to be wasted, and if not wasted and inadvertently used, you could have the risk of contamination. The second difference is how the bottle is actually sitting within the plastic packaging. As I said before, the instructions are to open the product and allow the bottle to drop into a sterile field. In this situation, the RLD freely drops when you peel away the packaging, but the ANDA does not. There's a groove there by the bottleneck that keeps it in place. This difference may compromise the sterility if the end users do not employ proper sterile technique during the bottle removal from the tray, which may require an additional manipulation that's not present with the RLD that drops freely. So with these examples and our experience with comparative analyses for ophthalmics, there's several lessons learned I want to end with. First, ophthalmic drugs make up 15 to 20% of the comparative analyses submitted in ANDAs. Considering the context of use of the product, ophthalmic drug device combination products are typically acute and chronic use and administered by the patient or caregiver. User tasks are self-evident, and products usually do not include instructions for use. The assessment of the user interface for ophthalmic drug device combination products require collaboration with the drug product team to ensure that design differences will not pose harm to patients. Thank you very much.
Good afternoon. I am Ajay Khopade, Vice President R&D and heading sterile non-solid oral dosage form formulation and Pharma, India. Today I'll discuss the challenges in developing complex generic ophthalmic products and how regulatory expectations influence our strategies. This session is a part of FDA CRCG Visionary Standards Workshop, which aims to advance science and regulation of ophthalmic generics. Over the next 20 minutes, I'll share practical insights and real FDA queries that highlight these challenges and the learnings gained from it. The presentation is a reflection of the experience, and today we'll not discuss the questions why, because we have already discussed that yesterday. But at the end of this presentation, I'll be sharing the industry's perspective where the guidance and clarity is needed. Before I want to clarify the views expressed here are my personal opinions and do not represent any official stance of Sun Pharmaceutical Industries Limited or its subsidiaries. So here is a roadmap for today's discussion. We'll start with the Q1, Q2 sameness, then move to Q3 sameness, which includes drug distribution, globule size, particle size, in vitro release, and redispersibility. We'll also cover unit dose sameness, manufacturing complexity, container closure system interactions, regulatory compliance needs like impurity control and particulate matter, and finally, bioequivalence strategies. These sections will include expectations from the queries to show how these issues play out in practice. So achieving Q1, Q2 sameness goes far beyond listing excipients. It requires matching grades, substitution patterns, and viscosity profiles for polymers like HPMC, PMC, or carbomer, as these govern rheology, redispersibility, and release. Regulatory transparency on the quantitative composition pushes sponsors to harmonize pH, osmolality, and viscosity with R&D capabilities. Trace excipients and processing aids demand ultra-sensitive specific methods with firm LOQ, LOD, and strict peak identification rules, which become particularly challenging when excipients are complex mixtures. Minor packaging differences can alter delivered dose under ideal conditions, not reflecting user variability. Thus, pairing composition with the drop size and unit dose uniformity evidence is difficult. Where formulation details remain undisclosed, sponsors must rely on science-based bridging using comparative data and risk-justified controls rather than assumptions, essential for compliance, yet operationally demanding. Q3 sameness challenges. From the industry perspective, achieving Q3 sameness from emulsions is highly complex because it hinges on the accurately locating the drug across oil, aqueous, or micellar domains. Determining API distribution between oil and aqueous phases in ophthalmic emulsions is critical for quality and bioequivalence. Yet validated methods such as ultracentrifugations or orthogonal techniques like ultrafiltration or non-invasive 19F NMR require significant expertise and resources. Challenges include achieving mass balance, ensuring method specificity to separate phases without artifacts, and avoiding phase perturbations during analysis. With no single standard regulatory protocol, sponsors rely on fit-for-purpose validations and recovery checks which may not fully satisfy expectations. Analytical windows must demonstrate robustness to manufacturing perturbations as homogenization pressure, passes, temperature, and include statistical plans that discriminate shifts from the critical material attributes and critical process parameters. Clear documentation linking development studies to master batch records conditions is essential to confirm exhibit and commercial processes preserve the validated Q3 state. Critical, yet operationally challenging. Globule size control in emulsions. From our perspective, precise control of globule size distribution is operationally demanding because regulators expect validated DLS or laser diffraction methods with correct refractive index, serial dilution SOPs, and setting consistent with the validations. Queries often require aligning the process GSD limits and final DP specification and correlating span or percentiles to CPPs, such as homogenization pressure, passes, temperature, and residence time. Population bioequivalence analysis must be supported by tight data based limits reflecting RLD mean plus 3 plus or minus standard deviation with representative histograms across dilutions and stability. Method system suitability should capture sensitivity to agglomerates, while sample preparation must avoid coalescence or breakup. Manufacturers must demonstrate scalability, pilot and commercial outcomes matching development space, and embed routine controls, hold times, rework limits that protect GSD during filtration, storage, and filling, preventing drift that could compromise dose uniformity and ocular tolerability. ESD control for suspension from industry perspective, maintaining B equivalent particle size distribution is highly complex due to milling and homogenization choices, dry versus wet, dispersant effects, and the variability across RLD lots. Regulatory queries favor robust validated image analysis or laser diffraction methods, microscopy correlation, explicit filter parameter justification, such as area, solidity, and agglomerate classification thresholds demonstrated using RLD. Acceptance criteria typically include D10, D50, D90, and sometimes upper cutoffs such as D95 and D100 to mitigate irritation risks. Sponsors must define CPP and PSD linkages such as media size, load, rotor speed, passes, or temperatures to show accuracy and precision over the realistic matrices. Sampling must prevent re-agglomeration or solubilization, and dispersant or sonication effects are the part of robustness. Population bioequivalence plan require adequate lot replication, and specifications should reflect the RLD variability while remaining discriminating enough to reject non-similar batches produced and purposeful parameter shifts, critical yet operationally demanding. For in vitro release, perspective is that ophthalmic emulsions and suspensions present as significant challenges because of the universally accepted biorelevant IVRT conditions do not exist. Regulated expects specifically justified membranes or filters with low non-specific adsorptions and multi-point profiles spanning early, mid, and late time points. Queries emphasize mass balance, drug plus oil wherever applicable to confirm the method measures release rather than droplet leakage. Demonstrating discriminatory power is critical, requiring evidence that method rejects batches with deliberate variations in viscosity, ESD or GSD, or composition. Early development and validation must incorporate specificity, robustness, and stability indicating elements, while preparation should mimic patient use such as shake well, sometimes limited to less than 30 seconds if it is labeled. When IVRT cannot be reliably bridged to clinical performance, sponsors must provide a surrogate attribute out of Q3 metric and justify its clinical relevance using comparative data. It is essential yet operationally demanding. Regarding redispersibility, from our perspective, redispersibility is pivotal for dose uniformity over shelf life, yet achieving it under regulatory expectations is highly demanding. And queries will typically focus on unbiased sampling from top, middle, and bottom of the distributing matrix, validating low volume assay precision and recovery and controls against sedimentation during the sampling or transfer is difficult. A combined approach is favored, the qualitative time to disperse plus quantitative layered assay within the RSD limits and the equivalent statistics against RLD. So sponsors must link the redispersity to viscosity, rheology, and PSD, evaluating orientation effects, upright, side, inverted, and include the in-use studies simulating realistic handling. Method SOPs must be detailed, must have detailed pipetting practices, anti-evaporation measures, and timing to minimize bias. Specification language should hit acceptance criteria for both release and stability with trend analysis to detect any shift and trigger investigation before the clinical risk emerges. Again, it is essential, yet operationally challenging. Regarding unit dose sameness, ensuring unit dose consistency in ophthalmics is highly challenging due to inherent variability in drop size, suspension, settling during dosing, and the influence of blotter and closure design. The regulatory expectations for the unbiased dosing procedures, avoiding priming bias, along with the orientation studies, multiple angles, at times at multiple angles, and the reporting of individual unit results typically across 10 samples impose a significant operational rigor. Acceptance ranges of 90 to 110% for suspensions demand tight process control. Sponsors must verify the maximum daily dose using measured drop volumes throughout bottle life, start, middle, and end, and assess temperature cycling effects, adding complexity to the stability programs. Analytical methods requiring stringent system suitability, evaporation safeguards, and the precise timing to prevent loss increases testing burden. And for combination product ergonomic differences affecting actuation force, the tip geometry must be justified or bridged through human factors data, further complicating the development. Specifications and control strategies need to link unit dose performance to GSD or PSD, viscosity, to ensure reproducibility from lab to commercial scale. It is also essential but resource intensive requirement for compliance and patient safety. There is a lot of manufacturing complexity associated with emulsions and suspensions. And from the industry standpoint, the complexity lies in meeting the stringent regulatory expectations while maintaining the operational feasibility. Multi-phase aseptic processing demands precise sequencing of additions, validated SIP, autoclave cycles, and the tight control of critical process parameters such as pH, viscosity, and particle size distribution. FDA expectation on holding time studies for both chemical and microbial risk at every intermediate stage adds significant analytical and documentation burden, ensuring bulk and fill uniformity, reconciling development conditions with the master batch record for exhibit batches, and executing media fills that replicate the real-world equipment environments, filtration, filling assemblies, and maximum run durations, including restarts and discard volumes, are resource intensive. Further yield reconciliation, pre and post-filter integrity testing, and aligning filling yield limits and the actual process capability require robust data systems and operational discipline. Demonstrating scalability and SUPAC readiness through data-driven ranges and lifecycle validation introduces additional complexity, especially when changes in the container closure systems or process steps occur. In such cases, we must provide manufacturability evidence such as surrogate batches, torque studies, and container closure integrity testing, confirm readiness for routine sterile operation. While these measures are essential for patient safety and compliance, they pose significant challenges in terms of cost, time, and technical educations for the industry. Regarding container closure system interactions, the container closure maintaining sterility assurance and dose consistency in ophthalmic container closure system is complex due to semi-permeable materials and multiple joints like tips, plugs, and caps. The regulatory preference for deterministic CCIT as per USP 1207 often require both vacuum and pressure methods as technical and cost burden, as these tests must detect micro leaks, and dislodge debris. Plastic deformation complicates the probability dry ingress. So sensitivity must be proven with the breached positives and growth promotion controls, increasing validation scope. Integrity demonstration across the shelf life and post-sterilization worst case demands extended studies. Balancing ease of opening with the sealed robustness is practical challenge. Torque windows must protect integrity yet remain patient friendly. Lifecycle validation expectations include visual package evaluation in specifications, further strain the resources. Any redesigns require manufacturability evidence and consistent CCIT performance under routine commercial conditions. Critical for compliance, but. but operationally demanding. Regarding extractable, leachable, from an industry perspective, implementing a comprehensive extractable and leachable programs is highly demanding due to the breadth of requirements and inherent and uncertainties associated with it. Defining an AET validating screening and targeted method with adequate LOD, LOQ, and performing worst-case simulations such as temperature, duration, orientation, freeze-thaw require significant resources. Ophthalmic systems involve LDP bottles, plugs, caps, liners, inks, adhesives, and manufacturing contact materials such as filters, tubings, greatly expanding the risk space. When screening suggests potential exposure above thresholds, targeted leachable studies on stability and toxicological qualification, including ocular and systemic considerations, add complexity and require clearer guidance. Polymer batch variability and colorants introduce uncertainty, making material certifications as per CFR, USP, and component traceability is essential, yet operationally challenging. Control strategies must correlate leachables to impurity specifications and stability monitoring, ensure no changes towards safety margins over shelf life and in use periods. However, given the notorious unpredictability of the leachables, achieving this consistently remains a significant difficulty despite of a regulatory necessity. There are general container crusher system challenges. Specialized ophthalmic container closure systems components pose significant challenges due to limited suppliers, long lead times, and stringent multi-region compliance requirements, creating scheduling and change control risks. Regulatory expectations include sterilization compatibility like ETO, gamma, moist heat, cold chain resilience where applicable, and the evidence of consistent drop delivery across orientations and bottle life. Design considerations such as tamper evidence with the positive retention, torque range balancing usability and integrity, the drop size control within 20 to 20 microliter, but within plus or minus 10% of the RLD, add complexity. Human factor requirements treat dispenser as a combination products, comparative analysis or targeted human factor studies may be needed when geometry or actuation forces differs. Ensuring label legibility for the elderly and usually impaired user remains difficult. Specification must embed visual package checks alongside weight loss test and the semi-permeable containers critical for compliance, yet again operationally demanding. So regarding the impurity controls, from the industry standpoint, ophthalmic face demanding impurity expectations driven by ocular safety concerns. Regulatory queries push for individual specified impurity limits, lower caps for unspecified impurity impurities such as often 1.1%, less than 0.1%, and inclusion of new predicted degradants, new or predicted degradants with structural confirmation, adds complexity to the analysis. Reconciling a drug substance to drug product carryover, aligning nomenclature to the compendia, USP, Pharma Europa, and capturing precursors in the drug substance specifications when drug product degradants appear are operationally challenging. When matching RLD profiles, example, different API routes, near expiry RLD comparisons, and toxicological assistance offer clarity, but preclinical toxicology-based limits need further guidance. Validation packages must demonstrate specifically during dilution integrity and tight system suitability for decision-making at low levels, which is resource-intensive. Stability programs must confirm these limits hold under manufacturing, shipping, and in-use conditions with trend-based justification for shelf life and any tightening at approval. Again, critical, yet difficult to execute consistently. Again, from industry standpoint, detecting vesicle particle matter in opaque or semi-permeable ophthalmic presentation is inherently difficult. The expectations for validated inspection strategies that distinguish formulation particles, example flocks, agglomerates from foreign matter, without compromising sterility, add complexity. When visual failures occur during stability, linking observation to the intrinsic behavior is challenging. And root cause investigations requiring control adjustment, for example, in case of milling parameter or viscosity modifiers, demand exhaustive efforts. Sponsors also struggle to justify sensitivity of the non-destructive methods, including operator training and the EQL-based programs, complemented by subjectivity in the microscopy characterization. Optimizing particulate and foreign matter controls in both release as well as stability specification, while accounting for the orientation and in-use effects through a risk-based approach remains operationally challenging. Ultimately, the goal of demonstrating patient-safe clarity while respecting formulation microstructure and container constraint is clear, yet achieving it consistently under a regulatory scrutiny is resource-intensive. Regarding stability, the ophthalmic stability programs are highly demanding due to humidity sensitivity of the semi-permeable control closure system. Orientation effects for suspensions and the need for in-use realism aligned with the labeling, such as 7 days post-opening, in-use study post 7 days opening. The regulatory expectations for ICH consistent protocols with intermediate conditions when accelerated shows plus or minus 5% assay change, inclusion of weight loss test and the trend-based data supporting shelf life adds significant complexity. For suspensions, long-term upright on the side, inverted studies to capture caking or redispersivity changes impose a heavy analytical burden. Where impurities arise, tightening limits or justifying it via near expiry RLD approaches is clear, but guidance on the usage-based toxicological assessments remain limited. If extractable and leachable risks exist, leachable monitoring under worst-case storage and qualification clarity when exceeded would help. Exhaustive in-use protocols, preferably on the exhibit and commercial lots, meeting current specifications, must evaluate unit dose uniformity, IVRT, IVR, redisposability, and microbiology under realistic handling. Yet the term realistic introduces ambiguity and the operational uncertainty. Regarding in vivo B requirements, from an industry standpoint, the lack of validated in vitro surrogates forces reliance on the clinical endpoint B, which is costly, time-intensive, and often poorly correlated to the Q3 attribute. While regulator encourage building the strongest possible in vitro sameness case through robust PSD-GSD control, discriminating IVRT and tight unit dose uniformity, developing a predictive ocular PK models as supportive evidence add complexity and yet requires specialized expertise. Clear IVIVC guidance would significantly influence investment decisions tied to trial feasibility and timelines, yet remains limited. Although pre-ANDA dialogue is promoted to de-risk programs, confidence in proposing attribute-based surrogates validated to reject purposeful process of the formulation shift is still minimal. Aligning population bioequivalence statistics to RLD variability further complicates the study design. Sponsors face the challenges of demonstrating therapeutic equivalence through a layered approach when direct, reliable in vitro proxies are unavailable. They are again essential regulatory requirements and expectations, but they are operationally demanding. So overall, from the industry perspective, industry seeks greater label transparency, quantitative excipients on RLD labels, compress development timelines, and reduce Q1, Q2 uncertainty. Clear SUPAC guidance tailored to emulsion suspensions would streamline lifecycle changes and the CPV. Sponsors also request acceptable alternatives for Q3 differences when Q1, Q2 are matched, including formal pathways for drug distribution studies in a multiphasic system and practical P options when implementation is challenged by the RLD variable. Additional needs include regulatory clarity on the particulate inspection for opaque containers, recognition of attribute-based surrogates or ocular PBPK to support BE waivers were justified. And consensus on drop size, dose delivery studies across orientation and bottle life. Overall, aligning the regulatory expectations and science-based practicality can accelerate patient access while maintaining the ophthalmic safety. So with that perspectives, closing my presentation. Thanks to US FDA team, CRCG team, University of Maryland and University of Michigan, and Pharma R&D team. Thank you for your patience.
Go anywhere yet, Ajay. We're going to convene our expert panel. Wasn't that a nice set of talks from the discussion on the drug-device combination products onto a holistic view from industry regarding some of the challenges and some of the concerns from a drug product manufacturer perspective for these types of products. I can have all the speakers, and then is Nate going to join us online? You know? Okay. When he does, you'll let us know. So, Nate will join us from St. Louis virtually. Andrew, Shinae, you're on? Yep, great. Everybody's here? All right, so the floor is open. 2 questions. Does anyone have any questions for our speakers regarding the talks that they just gave? Andre.
Yeah. Hi, good afternoon, everybody. Thank you so much for the great talks. I have a couple questions. My first question is for Dr. Ahmed. I actually thought your presentation was quite inspiring. And in terms of the hybrid approach that you have proposed here, we would definitely be interested in talking about the details of that through the pre-ANDA process. So it's definitely something that would fit the scope of a pre-ANDA meeting. So we welcome you to submit that. But I was wondering if for the appreciation of the audience here, if you could talk a little bit more about your hybrid approach, what you intend to do, and what are the potential advantages and disadvantages of that approach?
Thank you. I think this is a kind of a learning which we have gained for other products development, what we have seen, and we want to implement in this area. See, the challenge is if you have to do a full-fledged clinical endpoint study, the investment, the time, the variability, and we know the reason these IVRTs are coming because we know those are less sensitive. I think the hybrid approach which we want to see or we want to propose is when we are seeing some differences in the Q1, Q2, and obviously when we see the documents, it clearly says it's a very standard and a statement that you have to justify and say that those Q1, Q2 will not have any impact on the safety and efficacy. It's very good to read that statement, but how do you prove that? So in order to kind of prove that, this is kind of an additional weight of evidence which we want to put in a preclinical study and a bridging study, a small pilot study, and the reason I'm saying we want to propose this is because we had tried this in maybe in past in oral solids scenario where this kind of approach was successful, and we want to try it out here where it's a kind of a balance and we kind of show evidence in terms of safety and efficacy as well, that the minor differences which we are seeing, they don't have impact on the clinical outcome. This is one purpose. The second, what it will add is it will generate more data, and then the challenge which we are seeing in terms of the modeling and interspecies scaling, that will also be added to that. So this is the kind of thought process which we have.
I think one challenge they have with a small patient population in that study is you're going to have some uncertainty based on the variability of those patients. And then how do you build that into the totality of evidence? I think it may be very much product specific too. So any company that wants to pursue that kind of approach should come in and talk with us in a pre-development meeting, pre-submission, or a pre-development meeting would be ideal for that kind of discussion. And there'll be some risk-based understanding of the product and concerns that might raise.
Thank you. If I may have a second question, this is more general for the panel. In terms of these complex drug-device combination products for ophthalmic use, as we're moving towards newer implantable and insertable products, newer technologies, in which sometimes the device constituent part of the product and the drug constituent part of the product may be hard to define from the industry perspective, at least. For some of those products, we may not have a PSG out just yet that clearly identifies what is the device constituent part here, what is the drug constituent part. So I guess the question that I would have for you is, from a general perspective, how can industry start looking at these products, and what would be the general criteria that they should consider in order to determine what is the drug component in this product, what is the device component in this product, when they appear to be intertwined?
Okay.
So I briefly mentioned in my presentation. So implant and insert product is really product specific, whether this implant or insert is considered as a drug and device constituent part or not. The applicator is very clear. If it is co-packed with or preloaded in the applicator, then the product is obviously drug device combination product. However, if it is only provided with the insert or implant, the best choice will be to submit a control correspondence to clarify whether it is considered a drug device combination product or not. It's because our decision to make a device constituent part is very comprehensive based on the multiple factors we need to see. And we also consider the labeling in the RAD as well. So it's really product specific. So please submit if there is an uncertainty or the unclear things about the classification, please submit the pre-enter program.
And as you know, we are rapidly moving to include information about the device constituent in the product-specific guidances. There may be some inconsistencies as we move along and evolution of what is the device constituent, because there are products where the dropper tip, for example, was saying that's the device constituent, but as was pointed out during some of the presentations, there is an integral nature of the container as well, and the pliability, the stiffness of the walls of the container that may factor into how much drug is delivered. But it was also pointed out that the typical drop is a lot larger than the capacity of an eye. So you're basically, your dose dispense is more than what an eye can actually handle. So you're basically trying to reach the maximum delivery for it. Having a slightly less thing that you pointed out, like 8 microliters instead of 10 microliters, in comparison may not always fly. Like you'll probably want to at least apply as much as an eye hold if you're going to match the reference product.
Thank you.
Thank you very much for sharing from industry's perspective what you see as the remaining challenges. If I understand you correctly, for instance, how you view the motivations for potential hybrid, and that's something carries potential, scientific values for us to further discuss. But I was curious in terms of, if available, if you could share more in terms of what your either experienced or anticipated potential difficulty in terms of, characterization of the polymer, and what's your current understanding as for how you would consider, conclude on the polymer similarity from your perspective?
This is for the Sun Pharma?
I mean, I think both
Speaker?
probably
Okay.
have something to share.
Okay, so regarding polymer perspective, I think one of the examples which I would state is regarding carboxymethyl cellulose. So initially, whatever the supplier who is providing us, I think he gives us the specifications ranging from, say, about 48 CPS to 110 CPS. I'm just saying arbitrary. I don't exactly remember what is the range for a 2% solution, and that is how you qualify and develop a product. essentially while developing a product, you would match the viscosity and content. Some of the information based on patent, you finalize the composition, you get a Q1, Q2 sameness, and develop a product. But in meantime, what we experience that the manufacturer changes its location and perhaps changes its sterilization. I mean, their molecular weight control was through gamma irradiation. And during that period, actually, something changed, and consistently we got a viscosity which is on the higher side. So initially we were at about 60 CPS, and we were consistently getting that for a period of time. And then when they changed, of course, they declared about almost 3-4 months before that they will be changing the site. We asked the documents, we asked the samples, and when they gave the samples, they provided all the viscosities which were above 90 and 80. And that was reflecting in the viscosity range, and which was going just above the viscosity range which we got specifications on. And that actually caused the problem. I think that was a background. So what we see is that if the viscosity is matching, I mean, like if the viscosity is our critical quality attribute, what we are trying to do is we would actually reduce slightly the concentration of the CMC and then maintain the viscosity of the overall formulation. So that is what perhaps we would try to do and justify.
Okay, yeah, so I think there are multiple layers, right? So from your end, it sounds like it's more life cycle management of your excipient supplier or potential different variability in the raw material. But I think from agency's perspective, right, in your case, and I think we heard in yesterday's discussion, sometimes there is a struggle in terms of if you meet Q2, you may not match Q3. But if you match Q3, then you may be outside the Q2 range. But I think for those cases, we don't have general one-size-fits-all approach, right? But then for particular case, if there is scientific justification for either the Q2 aspect or the Q3 aspect, I think those are where, you know, we highly encourage further like early engagement and discussion before you may have to, you know, abandon the previous program or switch to a completely different vendor. So where those cases, I feel we highly encourage the scientific discussion to see what can be done to move the program further.
Yeah, sure. So I think I appreciate that. But then essentially eventually what we have to come to a conclusion that we have to work with the vendor to actually set up the irradiation limits so that we get the product what we want. Okay, so yeah, so for a long time, our development was on the direction which I just stated that, okay, let us decrease the quantity, how much it is affecting the viscosity, but then eventually we have to work out and then convince them to give us sufficient data and its effects of gamma irradiation to optimize their irradiation process to give us the final samples.
Thank you.
Next question. Hello?
So I have a question about patent protections in the case of drug device combination products. So for Ozurdex, I believe what happened is the patent for the product itself expired first, and then the patent for the applicator expired several years later. So I'm wondering if a generic company is in a situation where the patents for the product have expired, and they want to imitate that product, but then there's still a patent in force for the device, and so they might want to try to circumvent that patent and maybe redesign the device. So then I don't know how that would be regulated from the FDA, because now the device is significantly different. So I'm just wondering if you all could discuss that. How does that work out if there are some expired patents, but some patents in force on the device versus the drug?
And Commander Fine?
So from the industry perspective, I mean, I can share what we actually do usually. See, when the product patent expired, it's very clear that you have to actually match the quality. But when the device exists, I mean, our preference is to actually keep the device as same as possible and work around the device in such a way that the requirements, the user requirements are minimally changed. If that is not possible, essentially in the generic scenario, the product doesn't become viable. So I think that's the situation which comes in, because the uncertainty of doing user study and especially for the critical procedure, I mean, like intravitreal injections, I mean, like what will make the change, it's very difficult to guess and assess. We try to engage with FDA, of course, that is the case. But then even before that, if at our internal discussion itself, we find it challenging, then typically we don't go further. And then perhaps there is no other option but to wait for the patent expiry.
Yeah, just a note that during the pre-development process for communications with the agency, we do encourage our applicants to be transparent about the nature of their design specifications for the device constituent. If they're designing around the patent, then do indicate that in your application to us. We're interested in seeing that, and that would make a complete meeting package, as part of a complete meeting package for your meeting with us.
I think just want to add, I think kind of echo what just said, but I think it's a balance between the business and the uncertainty in the risk of approval. So when we are totally changing the device, I think the extent of the studies which need to be done, those have to be assessed. And if there is no clear way forward, in general, the generic industry would be hesitant to change the entire device. I mean, it could be a kind of a way around or minor changes which could still be justified, but if it's a totally different device, that's something which kind of prohibits, unless there is a so strong business case that they take that decision, but in general, it's a no.
In addition to that, there is another, actually, you're talking about a device patent expiry, which is long. But then in the cases where device patent expiry is long, but at the same time, with the news items, you see that even the innovator is changing the device during that expiry stage. So they are already working on a new device, I mean, and therefore it becomes additionally difficult for us to make that decision whether to actually develop a device even for, say, for example, to 2030 launch. Because if they come with the new device in 2030, then you have to again judge and see whether it was replaced for safety and efficacy reasons. For example, a small silicone piece coming out of your retention kind of a thing. Now, that makes us, I mean, it makes a very difficult conversation within internal discussions to decide what is going to happen, right? I mean, at that point of time, actually, we also not very clear that is it a right time to go to FDA, because perhaps no one knows that whether the device will be changed or not. And even if they have filed and they are working, perhaps that is not the information we are going to get.
And the other thing is, per the statute, the Hatch-Waxman Act, sponsors are encouraged to test the limits of patent validity. So paragraph 4 certification, et cetera. So you encourage generic drug companies to step up, say, okay, well, is this patent really valid in the context of this product?
Okay, thank you. That's all very, very interesting. So my follow-up question is, say if the brand name company reinvents the applicator to extend the patent protections like you were explaining, is it then possible for the generic company to develop their device based on the old patent, the one that's already expired?
So yeah, so that depends upon perhaps FDA can control. I mean, if that is not replaced because of the safety and efficacy reasons, then that is clear. But then, we have to actually reach out and make FDA to determine whether this is replaced based on the safety and efficacy. If not, then yes, we can at least make that decision fast.
Commander
So that is where FDA
did you want
can
to comment?
Thank you. So great questions. For the previous question, I agree with the comments before, and I just want to take that back to the framework. The comparative analysis framework allows you to understand what the differences are between a device you're proposing, whether it's different for business reasons, whether it's different because of patent protections. The starting point is always the comparative analysis, so you understand what those differences are. And as he alluded to, if they're minor, it may not be prohibited, but if there are other design differences, you may need additional information to support that change. For the RLD changes, again, these are a challenge that the agency kind of is in the same boat as generic industry is. We don't have control if the RLD changes its device constituent part. So, but ANDA submissions do need to be compared to the latest approved RLD device constituent parts. So there could be a couple scenarios. One is they replace the old device and replace with a new one. That poses some challenges, especially if that new device has new patents around it. But there are alternatives where they may propose alternative presentations or alternative device constituent parts where it boils down to what the rationale was for that change, just to ensure that it was not for safety or efficacy concerns. But the comparisons have to be made to the latest RLD approval. And there's lack of control over when changes occur and if they occur.
Right.
It does help for a generic company to have an understanding of the full life cycle of the reference product. To see, for example, were there recalls for that product, the original product? Were there any changes made in the device constituent due to safety concerns that might have been found because of those recalls? So these are all things that will pop up during our assessment, and also how generic companies choose to develop their product.
I think with patent law, one of the expectations is that the new patent, it's not excessively incremental. It has to be a real invention in terms of improving safety or efficacy.
Not obvious.
So, yeah, so if it's just some spurious new patent, then the generic company, that one of their strategies, I think that you mentioned, is you would challenge the validity of the patent of the new one.
There are patent attorneys that make a career of figuring out which patents are more easily contested. Like some things are, we were just discussing that some of the formulation patents are easily contested due to obvious. So there are some issues with how valid certain patents are in the context of the product.
Yeah, very interesting. Thank you.
Any other comments for our esteemed panel here? Does our panel have any comments? And Nate, you're online. Do you want to comment on the role of the laboratory over in St. Louis?
Yeah, thank you so much for including me on this panel as well. I think that this discussion is great. Yeah, we do a lot of testing in our laboratories. I'll kind of maybe throw a question at maybe Dr. Kim and Dr. Fine about comparative analysis and whether they see a need to expand that framework beyond just like physical comparison, task analysis and labels and so forth. Things like performance characteristics or things like that, and maybe how this comparative analysis would work with products that don't necessarily maybe products that are provided by a healthcare provider rather than used by a patient as well. So if they can maybe comment on that or give us an idea of how that would work.
So, no, great point. So Dr. Kim and I touched on the user interface comparison, which we use the comparative analysis to identify those differences. But other device performance characteristics and comparisons are always going to be part of that ANDA as well. Whether they're quality attributes, whether they're device performance handled by the Center for Devices, all of that is an additional piece, whether that product can be therapeutically equivalent to its RLD based on changes and making sure that the performance characteristics are meeting set standards and also comparative to each other. So that we didn't mention that because that's really not our piece of the assessment, but that is happening as well for any of these applications, any of these differences in device characteristics.
Nay?
I don't think there is something to add for here, but so yeah, so our comparative analysis is only focused on the user interface assessment. So by the combination with the other device performance tests or other biocompatibility biocompatibility test for the devices. We're gonna decide the overall approvability of the ANDAs. But the presentation in this session is only focused on the user interface for the device consumer part. So when the device is the other design differences, then we need to consider many of the factors. However, again, when you follow up the comparative analysis process, and if you find a minor design difference, even though device shape or looks like a different user interface perspective, it could be a minor design difference. Then you don't need to worry about that much if the device platform is different.
Andre, and then Yuan, might have a question from our virtual audience.
Yeah, my question will be really quick. I was just wondering, as you were discussing in terms of minor and other design differences, and how we see that, and if there is any distinction in how we see that, depending on how the product is intended to be used. So in terms of Ozurdex, it's really meant to be administered by a healthcare professional. Would we view these minor and other design differences in a different way if that product were to be administered by the end user, by the patient? Is there any distinction here?
So if both of us discuss how the context of use of the product factors into the assessment of differences and classification of those differences. So in general, healthcare administered products would have more allowable differences, just for lack of a better term, compared to the RLD, because the end users is known to be more familiar with different designs, kind of professionally trained to administer products in a variety of settings. That may change if it's an emergency use product. There's other characteristics of that context of use. But in general, we see more other design differences that don't necessarily need additional data or kind of human factors data or in vitro data for healthcare administered products, because the additional information is that this is a trained user who has a low risk for making any errors if using the ANDA product in place of an RLD.
And along those lines, is there a difference between RX products and OTC products as well?
Yeah. I also like to add one thing. Even though it's a healthcare professional use only product, and if there is other design difference, of course, in general, they have a more trained to use the product, so it will be easily accepted, some level of data design difference. However, depending on the indication and the user situation, if it is a different external design attribute, it can be confused during the surgery, or some level of the critical indication could be occurred. So we does not consider just one thing for who is the end user. We're gonna consider multiple layer, all the context of the use, whether it is acceptable or the design difference based on the product specific.
Yuan, virtual question?
Okay. A few virtual questions. The first is, does the agency require manufacturers of eye drops to provide uniformity of dose for multi-dose bottle with dropper tip? For example, documentation shows that 2.5 ml bottle of eye drops will provide 25 doses.
Can you repeat that question again? I didn't hear you. And can you raise the microphone closer to your mouth?
Okay. So they just want to know, whether those multi-dose bottles with drop tips, whether they need to provide a document show that such bottle can provide a certain drops.
Amanda, fine.
Certain number of drops.
I'm not gonna. I'll only comment. These are quality attributes. So I know there's oftentimes dose accuracy studies, as drop size evaluations. So those are part of the application, part of the comparison. I'm not going to pretend I know because I'm not a quality assessor, but I'm seeing some nods in the audience that is correct.
So maybe they can reach out to OPQ or.
Yeah, I think the main thing is that you want to look at the performance of the reference product, and you should have a comparable performance.
Actually, the Dr. Kim is already mentioned about in the, oh no, forgot in the, but there is in the quality size of the decision is not in the drop size and the multi-dose of the drop performance within the multiple angle within the 90-degree versus 45 angles within the multi-dose case. That is kind of the we measure it in the single drop versus it's a multiple drop, and then just the detect or in the check. But that is not in the comparative analysis.
So if I may just add to that, I would direct who asked that question to the guidance for industry on quality considerations for ophthalmic products. That guidance does outline what is the expectation in terms of multi-dose bottles, what is the drop size, what is the potential variability that we expect in the drop size, and if that drop size is higher than that threshold, what would they need to show? So let's say, for instance, if you're working on a multi-dose bottle, you do a drop size study, you see that the drop size is above, let's say, it's 10% higher than the drop size of the RLD, right? In that case, the expectation here is that you would need to show that that bottle, your bottle, can dispense the same number of doses as the RLD. So that's the general consideration in terms of that.
And one more add thing is in that drop size study is the most important thing is in the based on the drop size decision, they can calculate for the maximum daily dose.
Yeah. Sounds like there's quantitative data to submit to alleviate quality concerns.
Okay, the second question is, could the agency please indicate the status of the draft FDA guidance, which was released for public comments, quality consideration for topic of ophthalmic drug product? I believe that one is December 2023. So what's the current status for that draft guidance? And also, the second part is, are there any other relevant guidance on the development for ophthalmic drug other than PSGs?
The general response is draft guidances intend to reflect the agency's latest thinking on a specific issue. And so you should take that 2023 guidance document as reflecting our latest thinking. However, how it's applied to a specific area or product is, you should take that somewhat into consideration, but it's not binding. But you can come in for clarification with the agency if you have specific concerns or questions in a pre-end meeting to discuss with us. And the quality folks, our Office of Product Quality folks are very interested in participating in those meetings, or you can send in control correspondence with a specific question, and even if it's related to a draft guidance already published, they're happy to address those.
Okay. The next question actually is general comments. They just want to know, if there's any comments from agency to the multi-dose container closure system for the preservative-free drug product.
In use testing, OPQ?
Yeah,
I don't know exactly what he wants it, but my interpretation is kind of in some of the ophthalmic product is in the benzoic acid-free formulation compared with in the original form. Change it, but there is no specific for the non-preservative product for the multi-dose container closure systems.
Yeah, there's a relative risk, right? If you lose the preservative, you can raise microbiology.
Yeah, this is something that we see every now and then. There is a move towards removing preservatives from ophthalmic drops in general. So it's not uncommon for generic industry to come in, and propose a generic product based on a reference product, but that generic product will not have preservative while still trying to maintain that product in a multi-dose dropper. Historically, there are issues there in terms of the container closure system device design and how you are able to maintain the sterility of a unpreserved formulation in that type of system. As you're aware, there are multi-dose bottles on the market for preservative-free formulations. So it is something that is possible, but I would suggest that that be discussed through a pre-end meeting request so we can consider all of the intricacies of what would need to be done in order to show that that alternative bottle is still able to maintain the sterility of the formulation while also not introducing additional risks such as potential metal ions, which has been a general concern with respect to those types of bottles.
It's also changing the formulation potentially.
Yeah.
Who else? Do you have anything else? Okay. So with that, we're coming to a close. Does anyone in the panel want to have last comments? Our industry colleagues, thank you so much for your participation and your wonderful talks. Really appreciate that. And Shinae and Ajay, thank you so much. Your viewpoints on the drug device combination products.
Thank you.
All right. With that, we're going to close this panel session. And we're going to move on to closing remarks. I understand we have a special guest for closing remarks, which is different from our schedule. Dr. Rob Lionberger is going to give closing remarks for this session. Dr. Lionberger is both a Stanford and Princeton trained chemist, chemical engineer, and his PhD work was on colloidal suspensions, right? Chemistry and in that environment. So it's highly relevant to this, some of the discussions that have gone on here. Rob Lionberger is also the director of the Office of Research and Standards in the Office of Generic Drugs here in FDA. Rob?
Thanks, Markham. Thanks to all of you who attended and participated, and especially those to you who participated online at this workshop. I think this is a really good example of what we're trying to achieve in our collaboration with the Center for Research in Complex Generics. Have a really focused meeting on a generic drug development topic, bring together the experts from all different parts of FDA and the industry that's working in these areas to understand what the current state of the art is, how to make things work better, and how to move them forward. So we really appreciate, from that perspective, the people from the generic industry that participated in the planning committee, worked through the CRGC, share their feedback on what the challenges are. Again, our goal in the Office of Generic Drugs is to have robust access to competitive generic products in all different product categories. And you really saw that, especially in today's part of the workshop, really where you heard about some of the new ophthalmic implants and the pathways for generic competition for those products. But we also don't want to forget that, again, there's wide use of generic products. So even for simpler products like the ophthalmic solutions, heard a lot through this workshop on what the expectations were, some of the challenges that you might encounter with looking at today the device, but also looking at the formulations. Yesterday, we're happy to hear what some of the challenges are, what some of the ways that we can make our processes work better on that. I think you may have noted that just several days ago, FDA finalized our guidance on Q1, Q2 waivers for pH adjusters. Right, this can be very significant for ophthalmic solution product development. Makes it very clear that there are cases where, again, changes in pH adjusters can be allowed in generic products as a clear pathway for what you want to do. And I think that's part of the landmark changes in that. You also heard yesterday about updates to our product-specific guidances as we're trying to make it very clear what the efficient pathways for generic development for ophthalmic products. And so with that, I'd like to bring this meeting to a close. Again, thank all of the organizers, the speakers, the in-person participants who made this event possible. For those of you who are here in person, we will be continuing with some breakout session discussions, but for all of those online, I hope you enjoyed this workshop. Again, like all of our CRC workshops, in a few weeks, the recordings and slides will be available for you online to share with others who may have missed this event and who are active in this ophthalmic area of health. With that, thank you all very much.